Hey, Hammy here, back with part 2 of section 3.2 on cell structures, where we're continuing to look at more organelles or parts of the cell. The first one we want to look at is one of the more obvious things you see when you look inside the cell, and that would be the nucleus. Okay, it's usually close to the center of the cell. There you can see it kind of in the center of the cell. We saw these when we looked at cheek cells in lab. Okay, look kind of like little blue cornflakes. You could see the nucleus, these little blue dots inside the cell. The nucleus is where the DNA is located. Remember, the nucleus would only be in eukaryotes and not prokaryotes, because remember, prokaryotes don't have a nucleus. Okay, and that DNA is what is going to code for the protein. So we often call this the nucleus kind of like the command center. You'll notice if you look here at the structure of a nucleus, there are pores or openings where RNA is going to read the code on the DNA, and it can get in and out through these little pores out to the cytoplasm to make the proteins. Okay, this is a lipid bilayer. It's not zoomed in on it, but it would be similar to the cell membrane where it would have little phospholipids, and it would make up this membrane right here. Okay, so this is the nucleus, often in the center of the cell, control center, because it has all the DNA or the instructions for everything the cell needs to make and do. The second organelle we want to talk about is mitochondria. Mitochondria is where energy is made available for the cell. Oftentimes we call it the powerhouse of the cell, right? You learned that in elementary school. Okay, the powerhouse of the cell. Why? Because it is the site of cellular respiration, okay, which is going to take glucose, remember C6H12O6, and break it down into the energy currency of our cells, which is ATP, adenosine triphosphate. Okay, so that is happening inside of these little mitochondria, and places in our body that need a lot of energy, like our muscle cells. Okay, our muscle cells have lots and lots of mitochondria because they need lots and lots of ATP or energy to do their job. Now, interesting thing about mitochondria is there's this thing called the endosymbiotic theory, where mitochondria and chloroplasts, we'll talk about in the next video, they think that originally they had lived on their own and then were engulfed by the early eukaryotes, and they're like, hey, so here's, if you look down here, so here's a early eukaryote, kind of some DNA, and hey, look, our DNA is important, so we're going to enclose it in a nucleus, and then it starts folding the membrane in to get some of our endoplasmic reticulum and that kind of stuff, and then it engulfs or swallows an aerobic bacterium. So like a prokaryote that had learned to do complete oxidative breakdown of glucose and get a lot more energy than just fermentation. And the cell said, why don't we just keep the little buggers alive? Because they give off a lot of ATP. And so the cells took them in, kept them alive. We have some evidence for this. Mitochondria has its own DNA that can be passed through maternal or mother to mother, from mother to offspring in every generation. Mitochondria replicate themselves, make some of their own proteins, and they have a double membrane. They had their original membrane, and then when they were engulfed, so if you look down here, when they were engulfed, where this cell membrane pinched in, okay, that ended up sort of giving it an outer membrane. So there is some evidence that this may have happened long ago in the past. Okay, so mitochondria, remember, energy, powerhouse, cell, respiration, okay, breaking down glucose for energy. The next organelle is just right outside the nucleus. Okay, so here's the nucleus. This would be in the middle of the cell and kind of connected to that membrane. Oftentimes I like to think of the endoplasmic reticulum, or sometimes we call it the ER for short, not emergency room, endoplasmic reticulum, ER. Okay, we sometimes call these the highways of the cell. Okay, it's going to be where proteins are made, on rough ER. Why rough? Because I guess if scientists imagine if you could get in there and you could feel the endoplasmic reticulum, see all these dots, dot, 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 dot. Those little dots are ribosomes. Okay, so if you could feel it, it would feel rough. Okay, I don't know. You'd have to get on the magic school bus and try it. Okay, with Mrs. Frizzle. But it looks rough. So we call it rough ER. That is where there are ribosomes, which is where protein synthesis is going to happen. Okay, and then the proteins are put into the lumen or the space of the endoplasmic reticulum and are going to be transported eventually to the Golgi apparatus, which we'll talk about. There is smooth ER as well. Okay, right here would be smooth ER. Okay, smooth ER doesn't have ribosomes, and that's used to help make lipids. And sometimes in our cells, it's where certain substances are stored as well. Okay, what are those ribosomes you speak of, Mr. Hampshire? Well, the ribosomes that stud the rough ER, we sometimes call it RER, rough endoplasmic reticulum. Those little bumps are what we call ribosomes. Okay, there's actually a large subunit and a small subunit, and they're made up of RNA and some protein. Okay, and these little guys are the ones that are responsible for protein synthesis. And we get into our chapter on DNA and how DNA is made into proteins. We'll look more closely at these little guys and see how they do their work. They are found either all throughout the cytoplasm. Okay, so you can have these kind of free ribosomes. Sometimes there's a bunch of little ribosomes all together that we call polyribosomes, or sometimes they're attached to the endoplasmic reticulum, to the rough ER. Now remember, ribosomes are found in prokaryote and eukaryotic cells both. Okay, so these are the little protein factories building up all those enzymes and different proteins in our cells. Last organelle here is the Golgi apparatus. Okay, everyone say that, Golgi, not googly goggly. I hear all kinds of things. Golgi apparatus, or sometimes called the Golgi complex. Okay, the Golgi complex I often refer to as the packaging department. Okay, if you look at where it's at, here's the nucleus. Okay, here's the rough ER, the RER. Okay, the rough ER, where here's the ribosomes making the proteins. Okay, the proteins are going to leave in a little transport vesicle, are going to leave the endoplasmic reticulum, and right here is the Golgi apparatus. They're going to go through the Golgi apparatus. Okay, so it receives the proteins from the rough endoplasmic reticulum, modifies them. And you remember, proteins have simple at the ribosome, it's just the primary structure, just the amino acids all strung together. The Golgi apparatus then starts to twist it and clump it into the secondary, tertiary, and quaternary structure till you get that final sort of globular, three-dimensional shape of the protein. Because remember, form fits function, F cubed. The shape of that protein is important for what it does. Okay, that is modified and then packaged in the Golgi apparatus, and it's either shipped to other places in the cell, or it can prepare it for secretion. So that vesicle, after it goes through, the farmer and me like to think of these almost looks like flattened feed sacks, okay, or flattened saccules, okay? And you'll see that there is a cis face and a trans face. So the cis face is where the vesicle, the protein is going to enter, and as it moves from little area to area, and it goes through all these different levels, is where it's going to get modified and modified and modified. And when it gets to the end down here is when it's in its final structure, and it'll either be used in the cell, or it can be taken to the plasma membrane and secreted like a digestive enzyme into our stomach or small intestine. Okay, so this kind of wraps up this part of the organelles. On the next video, we're going to look at even more organelles, and some of them that are very specific to maybe plants versus animal cells found in one or the other.